Good morning, everybody. So, as the chairman said, I should perhaps just briefly introduce myself. So, my name is Rod Hubbard. I've been at the University of York forever, it seems. During the 1980s, I did a lot of work in graphics and modeling, so I have some experience of the techniques and the methods that you're talking about. During the 90s, got involved a lot in structure determinations of various proteins, and particularly looking at protein-ligand interactions of various sorts. And for the last 8 or 9 years, I've been working part of my time with the company that's now called Venalis, based in Cambridge in the UK. And it's the output from that work that I'm going to be discussing with you today. Because what we've got interested in is using fragment-based methods to find new hit leads and candidate compounds for various targets. And from an analysis of that work, one of the things I wanted to go through with you was to ask the question whether you can use those fragment screening methods to assess target druggability. And I think what this also does, is to help provide a useful data set against which to benchmark the computational methods as well. Now, when I submitted the abstract for this meeting, I thought it was going to be fairly straightforward. As I've gone through and analyzed our data in a little bit more detail, I think it's thrown up quite a lot of interesting things that I hadn't quite appreciated myself. So thank you very much for giving me the deadline for actually analyzing the data properly by coming to give this talk today. And a final comment, actually, on perhaps quite a lot of that very nice talk we've just heard. Beware of rules, because they're retrospective understanding of long-ago experiments. I'll work on the acronym, but that's what rules are, a retrospective understanding of long-ago experiments. I thought before I started, we should make sure we've got some definitions right, because I think it's very easy in these areas to misuse words, to misuse definitions. Druggability is a horrible word. It's a very ugly word. But in my book, druggability can apply across the whole of the drug discovery spectrum. It applies to your early target validation. Do you think you've got a biological rationale that drugging that particular target will have a biological impact? It comes down to the chemical validation. Can you actually find chemical hits? Can you find chemical matter that can be progressed? Can a drug-like candidate actually be generated? We come back to the biological validation again. Does that candidate actually work in the various disease models? You then got clinical validation, and then you've got market validation. And arguably, you don't know if a target's druggable until you get well into that market validation. However, for the point of view of today, what I'm going to be focusing on is that chemical validation and asking the question, can hits be found that can be progressed? The second area that I expect I should probably ensure we've got the right definitions for are some of the various methods and some of the tools that I'm going to be talking about. So this is very briefly what I'm going to talk about for the next 10, 15 minutes or so, is what are fragments, the different methods we've used for screening the fragments, so that you understand, if you like, what the genesis of the data we're going to be discussing. We'll look at the hit rates for different targets, and we'll look at the correlation that has with computed druggability for the structures of the targets. And then we'll go through where we found some of the discrepancies and what we've learned from that. So, to begin with then, quickly, what are fragments? Fragments are small compounds that will fit into a binding site. If you've got your target binding site here, if you screen that target against small molecular compounds, you can find predominantly compounds that bind into the center of the binding site, occasionally ones that fit out on the outside. And the challenge in drug discovery is then to take those fragments and to either grow or merge them together to make hit compounds that can then be taken forward into optimization. As I'm sure you're all aware, the